conference meeting, and it's you know, and it's a fantastic pleasure to introduce the keynote speaker, Ramon Parsons, who will all be known to you since in 1997 he was the first person to discover PTEN and published a fantastic science paper that really opened new ground, which I understand has over 3,000 citations now. It's the most highly cited paper by a long way in the PTEN signaling field, and it opened up a whole new area of research, which none of us would be here probably without this pioneering work potentially. So I've been told by Susan to be very strict with time, to keep the talks to to time, and questions to 5 minutes for each talk. And Ramon will have 40 minutes, and I don't have a title for your talk, but I presume it's going to be something about PTEN.
Yes, exactly. That's exactly right. Well, I'd like to first thank you, Dario, and also thank you for all the organizers for inviting me and giving me this opportunity. Basically, I just did a PubMed search to see what are the number of papers per year published on with the PTEN search, and it's we're at about 1,200 for 2013. So it's actually climbing. So it appears to be a good business. Either that, or it's getting easier to publish. One way or the other. So I've moved recently, about a year ago, to the Icahn School of Medicine at Mount Sinai. This is the new building we're in. We're actively recruiting at multiple levels, if you're interested at all, in moving to the Upper East Side of Manhattan near Central Park. Please give me an email at the assistant professor, associate, full, postdoc, etc., graduate students. So anyway, to start out, the PTEN story, from my point of view, starts out with a paper work from Daryl Bigner at Duke University, who was doing karyotyping of glioblastoma multiforme, and identified that very commonly one copy of chromosome 10 was missing in the tumor chromosomes. And this led to a hypothesis. This was around the period of time when the RB tumor suppressor was identified, and this led to the hypothesis that there might be another lurking tumor suppressor on chromosome 10. And again, from my point of view, it was clear from the identification of the RB gene and other tumor suppressor genes that the mapping of homozygous deletions in cancer was a really useful tool for discovering new tumor suppressor genes at the time, because this is a pre-genomic era. And we started to focus on chromosome 10q23, and in fact, Mike Wigler's lab was right across the street here when we were working with him on this at the time, and we were finding a common region of loss of heterozygosity that was occurring in breast cancer that was focused on just one small band on the Q arm. And then Karis-Eng discovered through linkage analysis the syndrome called Cowden's disease was present within a very small interval of about 5 centimorgans. And then working again with Mike Wigler, one of the probes for subtraction hybridization probes that called RDA for representational difference analysis was mapping within the Cowden locus. So with that information, one of my postdocs screened all the markers that were available in this region at the time and identified one marker, here shown here, this one with this long name ending in WG9, that was missing in 2 different cases of breast cancer. And interestingly enough, in hindsight, we now know that those 2 cases were BRCA1 mutant breast cancers. Anyway, using that probe, we screened a back library and identified a bacterial artificial chromosome shown here, about 160 kilobases. And we then cut that up into small pieces and sequenced little bits of it and tried to order them as much as we could. And we identified multiple probes now that were missing. And here's a southern blot using one of those probes showing it was missing in brain tumors and in those original breast cancer cases that we showed before. And then we took that same back DNA and we cloned it into, it's a very old slide because no one uses this technology anymore, I don't think. We cloned it into the genomic DNA from that bacterial artificial chromosome into something called a exon trapping vector, which basically is a little mini gene, and you can put a new exon in it, and then when you do RT-PCR, you can then clone and sequence these products and then identify trapped exons. And 4 exons were trapped from this back, and 2 of them mapped to EST clusters that we could find by blast searches. So people had been sequencing cDNAs at the time, and 2 of these exons matched to these large number of ESTs, express sequence tag sequences by the search. And then using that, we assembled a contig and could show by RT-PCR that this was expressed widely. And then that one of those first 2 exons that we found, we can in fact found a tumor that had an intraexonic deletion. That was our first clue that we were really onto something, because up till then, a homozygous deletion, you don't know really for sure if you're looking at this at the right target. And then looking at the sequence, it was very interesting. It encoded a tyrosine phosphatase homolog with a very strong consensus sequence for the tyrosine phosphatase family, with homology to tensin and also to auxilin, and that's shown here. And those are signaling proteins without actual phosphatase activity, but with the phosphatase domain motif. And then when we placed these different exons that we identified, further refining this mapping, you can see we had a variety of intragenic homozygous deletions and different kinds of cancer, from breast cancer, glioblastoma, and prostate cancer. And this was one of the first lines we sequenced and found a mutation. This is a 2 base pair deletion in exon 1 that leads to a frameshift mutation in the cell line LN-CAP. And this is a variety of primary tumors in which, from the GBM cases that we looked at, and you can see this is old-fashioned data. You can see a lot of different mutations here. If you just look, each of these panels has 1 mutation in it. Some of them are harder to see than others. And they produced either frameshift, nonsense, or missense changes. And this led to this paper that we published that Dario alluded to with Mike Wigler and a variety of other colleagues. At the same time, Peter Steck and Sean Tavtigian from MD Anderson and also Myriad Genetics identified the same gene. They called it MAC1 for mutated in multiple advanced cancers. And soon after, we teamed up with Pik3ca to sequence some of her kindreds that were linked to chromosome 10q. And we found I think all of them, mutations, germline mutations in PTEN. A couple of interesting, 2 of the families had the same glycine to glutamate mutation that later we learned from several groups, including Icahn, that led to just a lipid phosphatase inactivation, but was still competent as a protein phosphatase. And so this was our early map where we had a bunch of missense mutations clustering in the phosphatase domain, and a series of frameshift and truncation mutations, and with the blue mutations being the germline and the black ones being the somatic ones from the different cancers we were looking at. And then quickly became clear that some sporadic cancers of a variety of types had mutations in PTEN, and in addition to Cowden's disease, the Benet-Zanona, which is a similar disease, but a pediatric variety of Cowden's disease. And actually, subsequently, Keras has gone on to show that a variety of different hamartoma syndromes are associated with PTEN mutation. And anyway, so the first phosphatase assay, well, a couple of different people showed phosphatase activity, but this is one of the very early assays showing tyrosine phosphatase activity from Nick Tonks's group and using a protein substrate. And then an antibody that Tonks raised and gave to us at the time was the first antibody we could detect the protein in a variety of different settings. Here's just a series of breast cancer cell lines, some with mutations, frameshift mutations. These 2 had missense mutations, and these were wild type, the ones that had the higher level of expression. Oh, there's a colon line, 2 colon lines over here too. So one of the very first experiments we did before we knew anything about what pathway the gene could be on was we expressed PTEN in an episomal vector in these mutant cell lines and also some wild type cell lines. And also this cell line that's got a T antigen transformed mammary cell line. And we could see that the colony formation was much reduced when we added PTEN versus vector controls, and even also versus a catalytically inactive phosphatase, so a mutant that just blocks phosphatase activity. And it was interesting, now also in 2020 hindsight, these 2 lines that were very sensitive to PTEN turn out they have Pik3ca mutations. So, and this line, was the only one that we screened at the time that wasn't responding to colony formation due to PTEN. And for many of these lines, we could see that putting in PTEN could lead to cell death. We also, with Ken Yamada, looked at the other phenotypes, and we found that PTEN could inhibit cell migration quite nicely. This is just an inducible system where you're inducing either the expression of the gene in 3T3 cells or an antisense vector, and a variety of experiments went into this showing that you could inhibit cell migration, and it turned out that mutations that blocked phosphatase activity could still inhibit migration, which has been confirmed by many other groups subsequently. And then we and others, Pier Paolo and TAC-MAX group and our group, embarked to test the hypothesis this was a real tumor suppressor by mutating the gene in the germline in mice. And in our hands, they got tumors, as pretty much everybody had a similar spectrum of cancer development. Here was one of the first polyps we found. This was Carlos Cardoncardo did a stain for Ki67, and it's very poor stain, but you can see this is picking up positive proliferating cells in this early prostate lesion. And when we just sort of did a pathology analysis on the mice, there were just many different kinds of tissues in the animals that had some kind of neoplastic transformation. It wasn't usually invasive cancer, but it was some kind of early malignancy of some kind. Some of them had 2 hits, some of them didn't. And then it was around this time when we were trying to do all this that Jack Dixon published the paper. This is the cartoon he had at the end of the paper demonstrating that PTEN was an effective lipid phosphatase that was able to dephosphorylate the D3 position of phosphatidylinositol 3,4,5 trisphosphate, and really and basically when it does this, it was able to shut off downstream signaling to PKB AKT and affect cell responses. And so we and many others tried to see if we could confirm this or not, and this is just some data I won't go into, just showing that one of the first plots where we could see PTEN affecting phospho-AKT at 473 when we added it to cells, and then this is just showing that we could reproduce that, and that we could in fact see PIP3 levels changing in cells when we added PTEN to them, and that we could see feedback activation of the insulin receptor pathway, which made a lot of sense given that this pathway it was shown in Drosophila and also C. elegans around this time, that PTEN was on the classic PI3 signaling pathway that's conserved in those species. And so we're seeing this kind of feedback regulation. This is just a northern blot showing upregulation of IRS2. When you add PTEN, adenoviral-delivered PTEN, you can see the infection causes a very strong upregulation of the transcript, and also the protein, and also the IRS2 P85 complex. And then when we started to have a little faith that this signaling pathway was actually involved in cancer, we started seeing if we could stain these PTEN mutant tumors that we were seeing for PTEN and also phospho-AKT. And you can see this is another prostate lesion. You can see the cells piling up on top of each other, stained for PTEN, and you can see that there's clearly a loss of PTEN expression in the lesion, and that this is really mirrored beautifully by activation of surface staining, cell surface staining for phospho-AKT, very nicely. This is in the uterus, showing a downregulation of PTEN in the uterus and activation of AKT in the neoplastic region. And this is just a BRDU in the same type of a zone, showing that there's increased proliferation in this region with low PTEN and high AKT. And this led us to hypothesize that potentially we could try to target some place in this pathway to actually have some kind of therapeutic result in our mice. And there weren't that many options available at the time that we could give in vivo. And so we were able to get a rapalog from Wyeth, a company that now has been merged with some other company at this point. And we were able to then see if, in fact, we could inhibit at the time we didn't know it was the Raptor-mTOR complex, and we just knew it was a mTOR complex, and see if we could actually inhibit. And we saw we were able to inhibit S6 kinase, work that we did with Charles Sawyers, and we also saw we could reduce proliferation and not affect phospho-AKT and block tumor progression. So this was back in 2001. So fast forward, that's sort of the really history lesson that I wanted to go over. It's been a long time since all those things happened, but I think it's kind of exciting that in some way rapalogs have entered the clinic and are useful therapy. So, and that people are trying to use a targeted therapy for PI3 kinase pathway-driven tumors, although that's still a work in progress. Anyway, last year we published, Ben Hopkins, a graduate student in my lab, identified with a prior graduate student, Barry Fine, that the message for PTEN had an alternatively translated open reading frame in addition to the classic PTEN, that's 403 amino acids. And this unique region had several interesting features. It was predicted to have a secretion signal and also a polybasic region. So I'm not going to go through the experiments because they're published this past year, but basically he was able to show that the protein can be secreted, it can be found in serum, and that it can use its polybasic sequence to re-enter cells and actually cause tumor shrinkage when they're xenografted into mice or allografted into mice, when tumors are allografted into mice. And the ones that were sensitive that we measured were PTEN deficient tumors that we knew were quite sensitive to PTEN from in vitro studies. And then we also have been working on two other proteins, one where we've noticed that NFIL3 can block FOXO transcriptional factor output, not in all targets, but in a subset of targets that can basically restrain FOXO's ability to induce apoptosis. And also, we found a P-REX2 is a PTEN interacting protein that can inhibit P-REX2. P-REX2 can inhibit P10 lipid phosphatase activity. And when we knocked this out and looked, and this is, I believe, MEFS, we could see that there was a downregulation of insulin-activated signaling downstream. And when we did this looking in response to EGF or PDGF in these cells, we saw no difference in the signal. And this was published early this year by another graduate student, Cindy Hodakowski, in PNAS in January. So anyway, so I'm going to talk about, I went really fast. Okay, so I'm doing fine. So I can slow down a little bit now. So, so basically, we wanted to start to explore the possibility, since P-REX2 can bind to P10, we wanted to explore the possibility that P10 could regulate P-REX2. I've talked about, and we published in prior meetings, the ability of P10 to be inhibited by P-REX2, but we thought that potentially, since P-REX2 is a RAC-GEF, and it's been previously shown to be able to stimulate cell migration, we thought it might be interesting to explore this relationship. And so we started doing this by focusing in on the C2 tail region of P10, which has been long known to be a region of P10 that can inhibit cell migration. And we asked whether if we use this, this portion of P10, if this could be occurring, it could be inhibiting migration, and it could, and if it could do this through the dependency on P-REX2. And so you can see here, one of the things that we saw while we were doing the study is that actually the C2 tail binds to P-REX2 much better than full-length P10 does on its own. And then we can start making mutations that truncate pieces of the tail over here, and this interaction is reduced quite a bit. And you can see when we transfect in P-REX2, we can stimulate migration. This is in BT549 cells, which are P10 mutant and express sort of moderate to low levels of P-REX2. And we can inhibit their migration with C2 tail, and we can really reduce the ability of this to occur with some of these different truncating mutants. We can also see that when you co-express the C2 tail region of P10 with P-REX2, we can see co-localization at the plasma membrane. And so it doesn't suggest that we're sort of sequestering the protein from the membrane. But at the same time, we thought, well, maybe it's having really a direct activity on the actually guanine nucleotide exchange enzyme capacity of P-REX2. So P-REX2 has got this DH-PH domain here that has the GEF activity. It's got two DEP and two PTZ domains and an IP4P domain in the long isoform that we study, that we showed originally to be the one that interacted and inhibited PTEN quite well. And prior work showed that, in fact, this is published in the PNAS paper, that the tail PTZ binding domain can interact with the IP4P domain, and that the C2 domain can interact with the DH-PH domain. And so we just thought, well, maybe if we just take this C2 tail region and mix it in a GEF assay, pre-incubated in a GEF assay, we could see an effect on GEF activity. And here's using MET GDP fluorescence. And you can see this is the change in signal when you have PRX2A in the presence of excess GTP. And then you can see how if we add C2 tail, we can effectively block, this is in green, block this signal. And you can see we change the slope. Here's the p-value. We've done it many times, and it's quite convincing. We also looked to see if mutations in PRX2, missense mutations in PRX2, could have an effect on this activity. And we showed that, again, the wild type could inhibit migration. I'm sorry, the wild type, the PRX2 wild type could be inhibited by C2 tail quite nicely, but these three different mutants were not well inhibited. Sorry about that. I mean, this, basically, it's the same as this. It's just another mutant. And when we looked at this, what could be the basis for this, we could see that one of the mutants, shown here, this 844D, basically binds quite poorly to PTEN, while the other two bound quite well. So then we asked whether, I don't have the data here because I thought I was going to run out of time. We thought we were going to, we actually asked in the GEF assay, could they behave, and they were defective. They're defective in inhibiting the GEF activity. Well, they're defective in the sense that a C2 tail cannot inhibit their GEF activity. So, so this is all the work. This story I'm talking about is work of a postdoc in the lab, Sarah Mency, and she's going to be giving a poster on this. So if I'm unclear, she can talk about it then. But basically, when we started looking at this in actual tumor biopsies and getting lysates from these, we were able to get also some normal breast tissue and also some prophylactic mastectomy. And this is one of the graduate students in the lab has been really active in getting these samples. And basically what we found is that there's a lot more PRX2A, which is the long form, that's this upper band here, in most breast cancers. And there's even more of this PRX2C, which is basically a very similar gene, but it's missing this IP4P domain, which contains one of the PTEN docking sites. And so basically what we saw is that we said, well, there's a lot of this protein here. Maybe it's in excess. So we did a GST or versus GST P10 pull down with these lysates to see what was happening. And in fact, we could see that the pull down preferentially occurs with PRX2A, as we would have predicted. And the interaction with PRX2C is quite a bit weaker. You can see it. It's right here. And this here's the input for these pull downs. And these are in ER positive and ER negative breast cancers. I also took out data again. I don't know. Here we showed NPRX2. We didn't have an exact construct for PRX2C, so we took NPRX2, which is sort of our poor man's construct for that. And we, in fact, see that this can stimulate migration, as you'd expect, because we know that DHPH alone is able to stimulate migration. But we also saw that a P10 C2 tail is not able to inhibit migration well with this. So when we look at this, we sort of have this model going on that we're discussing in the lab, that in the setting of a tumor that's got P10, high P10 and high PRX2, the PRX2 can interact via two interfaces to really nicely dock and inhibit P10 quite nicely, which can elevate phospho-AKT. But if the P10 is at equal molar ratios, it can inhibit PRX2 as well and can sort of not stimulate this RAC-GEF activity. If we have a situation where PRX2 is mutant and P10 is wild type, we can have a setting where basically, again, and I don't show this data, we can see this elevation of AKT phosphorylation when these are co-transfected together. But basically in this setting, what we're thinking is happening is we're able to activate invasion and migration through basically now P10 in this setting cannot inhibit PREX2A's GEF activity. And then finally, in the setting when you're overexpressing PREX2A and PREX2C isoforms, different spliced forms of the same gene, we think one potentially could function to inhibit P10, the full-length one, and that PREX2C, which doesn't seem to be, we think, regulated by P10, could be activating RAC quite nicely and stimulating migration. So that's kind of our model for this regulation, P10 regulation of PREX2. And again, there's more details and some very nice other data, I think, in the poster that Sarah is going to be showing. So to switch gears a little bit, another postdoc in the lab is also here, has been working on for quite hard for many years the understanding of Pik3ca mutations and trying to model this in a mouse model, and then also trying to identify a tumor system based on this oncogene that can be somehow valuable for understanding how do we better treat breast cancer. And so the initial studies were to generate knock-in mutations for E545K and H1047R. Oh, I should, let me just for those of you who don't know this cartoon, I'm sorry. PI3 kinase is mutated, Pik3ca alpha is mutated very commonly in breast cancer. It's got a hotspot mutation in the kinase domain and another hotspot mutation in this accessory domain right over here. There's many other mutations that occur, but these are the two main areas where you see mutations. They tend to activate the signal, and they've been shown by many groups to be transforming. In our hands, they are transforming on their own when they knock them in the mammary gland and activate them with WAP-CRE. So it's a lock-stop blocks activate with a CRE driver to activate the oncogene in the mice. And we can see that the females do develop these very small mammary lesions that are really very wimpy. And then what we then did is we combined them with other oncogenes, and one of them is MYC. And we can see this is MYC only. This is the Kaplan-Meier curve for MYC. And you can see in combination with MYC, PI3 kinase, or P10, or this one here is P10 het, are very equivalent in their ability to cooperate with MYC very nicely in these tumors. And in fact, when we look at the MYC tumors on their own without any oncogenes, we see downregulation of P10 message and protein when we do immunohistochemistry. This is kind of washed out, so it's really hard to see here. But this is just some of the histology. And one thing I want to point out is we've generated cell lines from these tumors. One of these lines is a MYC P10 null tumor, and another is a MYC Pik3ca tumor. And we can xenotransplant this, and we see very similar signaling and histology as with the primary tumor. And so working with Stand Up to Cancer's PI3 kinase dream team with Lou, we got all these different drugs that were targeting the PI3 kinase pathway, or even you could say more broadly, the whole tyrosine kinase signaling pathway. And we tried to kill these tumor cell lines over and over with different doses of different drugs. And this is just a dose response for GDC0941, which we like. It's a very effective inhibitor of PI3 kinase alpha and beta. And you can see it doesn't do a very good job inhibiting the growth, as did really none of the other drugs that we looked at. And then we JQ1 was the hot drug in the last few years. So we thought, well, this is a MYC-driven tumor that we're making. Maybe we'll see some effect with that. And of course, no, we didn't say anything. But what we saw was, in combination, a very nice effect where we could see very clear. This is the untreated for both, just like these over here with just vehicle. And this is just, you can see the lowest dose. In this case, we're using 1 micromolar of GDC and a dose escalation of, 1 micromolar of GDC0941, the PI3 kinase inhibitor, and a dose escalation from, it's cut off, 0.125 micromolar all the way out to 8 micromolar for JQ1. And so these are all going out to 8. So this is quite a nice result. We were very excited about this, and this is the quantification of the result over here. And you can actually see at many doses, actually the stasis or reduced cell number in our in vitro growth assays. And when we look at phospho-AKT, you can see here that JQ1 doesn't seem to change it, as kind of as you expect. GDC shuts it off quite nicely at 4 hours. And then we also see over here that MYC with JQ1 doesn't seem to change at 4 hours. And we do see, though, at 24 hours, relatively reduced MYC with either drug alone, actually, relative to the control, and that the combination, we're always seeing a nice reduction in MYC levels, and we can keep this phospho-AKT down. Anyway, so this is just looking at what's going on from a kinetic point of view. And one of the things that we noticed that was kind of pretty striking was that when we, this is well described by many people. I think Neil Rosen really has championed this feedback that you see here, where you inhibit the pathway and eventually you start to restore the signal. This is an hour, so this is out to 32 hours. And you can see here for phosphofoxo, we get quite high. We get almost to where we started with phospho-GSK3 beta. Similarly, PRAS40 also, phosphopras40. Other signals, not so much. And you can also see this upregulation of receptors, like insulin receptor. This is the beta subunit, and here's the IGF receptor, for example. And however, the combination, one of the things that we noticed is you basically, the JQ1 is able to keep the feedback down so that you can reduce the amplitude. It's not gone altogether, but it's quite reduced. And here for phosphofoxo, phospho-GSK3 beta, PRAS40, this is, you actually are just really shutting down this phospho-S6. This is MET. I forgot to talk about MET. MET also is quite a bit down here. And again, you can see reduction of insulin receptor. It didn't seem to do as well for IGF-1R. And this is the other cell line that's very similar, but also a MYC line, but also, I forget, I think this is the one that's PTEN null. And we see a similar pattern with clearly a reduction for many of these markers in the feedback activation of the pathway when we're using JQ1 in combination, which is the BRD4 inhibitor in combination with the Pik3ca inhibitor. So here's, in this case, HER3 got upregulated, so we show this panel here. You can see insulin receptor, you can see IGF-R, again, and finally, yeah, and we can also see in terms of MYC, you can see MYC levels are, they're not way down, but they're staying down here in this combination at this dose, but not as well as one would like, actually. So anyway, so here we tried another drug. So we tried in vivo JQ1, but JQ1 in vivo wasn't able to lower MYC at all in vivo. So we just sort of said, you know, this is just, we can't deliver this drug at enough level to be able to ask the question. So we were able to get another, basically another BRD4 antagonist or inhibitor, whatever you want to call it, that from Mingming Zhao at Mount Sinai. And he's been doing work with it in vivo, and so we were able to use it in combination. And so this is xenograft data with the vehicle, this other BRD4 inhibitor, the GDC, and then this is the combination. And this is the data for, this is the tumors taken out from the animals, and you can see clearly these are the two different cell lines, one in each flank, and you can see that the combination is clearly better than either drug alone. And here's some of the signaling data we're seeing from the combination in vivo, and you can see that we're really turning down a MYC and AKT, and we're seeing evidence of cell death here. So that's basically that. So again, Elias is going to be presenting his work here at the meeting in a poster. So if you have any questions for him, he'll be happy to answer them. We're hopefully preparing this paper soon for submission. Anyway, so I'd like to acknowledge the lab and all the people that have been doing the work. These are the current members of the lab here. Many of them are here at the meeting. The people's work that I talked about was Ben Hopkins a little bit, Sarah Mency, Elias Stratokopoulos, and also these are some former lab members that are near and dear. Cindy Hadakowski just recently left and is now still in Manhattan, and but she's gone on to do a nice postdoc. And then these other former lab members and many collaborators that we've been working with, pathologists that and cytogeneticists that have been helping us over the years, bioinformaticists, Carlos Cardoncardo, and some other collaborators working on some stuff I didn't really get a chance to talk about. Anyway, thank you very much.
Thank you for finishing with 5 minutes and 25 seconds to spare. So my suggestion is we have a 10-minute discussion rather than a 5-minute discussion. So does anyone have any questions? And don't be shy.
Have you had a chance to look at resistance mutations or that would happen in the combination therapy?
You know, I think that's a really interesting question. We haven't done that yet. We should. It would be a really interesting thing to see, particularly in vitro. We get such a striking result in vitro that it should be doable. Good question, but don't have any data.
Ramon here.
Yeah.
Okay, so Ramon, so you showed the PTEN kinase inhibitor treatment induce the AKT reactivation, so that has also been shown by other studies.
Yeah.
Then you show the combined PTEN kinase inhibitor with JQ1 can abolish the reactivation of AKT activation. So can you comment the mechanism? Because one would expect maybe the PTEN kinase inhibitor treatment actually can induce MYC activation, so that can explain. So when you combine the JQ1 with your PTEN kinase inhibitor, so that can abolish the reactivation. But it seems like the PTEN kinase inhibitor treatment does not affect or maybe even decrease the MYC activity or expression.
It definitely, can only say what we're seeing. I mean, we're definitely seeing no upregulation of MYC with the PTEN kinase inhibitor at the protein level. At the mRNA level, I'm not sure what's happening. I'd have to ask Elias. Elias, do you? How does, in your model, how does the PTEN kinase inhibitor affect the mRNA level of MYC? Feedback upregulation? Yeah, it goes up. The mRNA is going up, but the protein level is going down.
Nice.
I had Ramon here. Great talk. I had a similar question regarding this, the question that you got now. And is the MYC effect seen in each and every cell lines, or it's cell line specific?
In our model, everything seems to be really good. We've invented our own little universe of this MMTV MYC model. Seems to be very nice and consistent. When we leave that universe and go into real human tumors, we see other things. Basically, actually Mingming Zhao just published a paper with some collaborators showing that in triple-negative breast cancer, which often have MYC upregulation and are missing PTEN and have sort of this combination that we're seeing, that they basically respond to JQ1 on their own. And he's just published this paper in Cancer Cells saying it's through regulation, I believe, twist, transcription activation. And what we've seen is when we've screened these cell lines, almost all of them are very sensitive to JQ1 on their own. So we don't have to inhibit the PTEN kinase pathway. So we couldn't do the comparative study. So we did find one cell line, however, human cell line, that has the same sort of properties that we're seeing. It's also a breast cancer line that has a PTEN kinase mutation and overexpresses MYC that seems to have behaved like these tumors. So it's complicated for sure. But it's interesting that we can actually find a combination that targets MYC with PTEN kinase in our model that seems to have some benefit. So my guess is the problem may be that BRD4 is doing other things in other systems beyond MYC that are very important for those cells to grow. So obviously, a better, more focused MYC targeting would be quite important, and we don't have that in our disposal.
Two other quick questions. The toxicity of the combo, Any comment on that?
It's not. We see weight loss in the mice with the combo. So we do see. We had to stop the study because of weight loss. That said, we haven't done any optimization of delivery to try to figure out if we gave them a drug holiday for a day or two, or we've just been giving it continuous when we give it in vivo.
Going back to the secreted PTEN, which I think it's an exciting data and finding, do you think that this could reconcile the fact that in certain tumors PTEN has been found to be lost in the stroma? Do you really think that loss of PTEN in the stroma can drive tumorigenesis in the epithelium because the stroma cannot feed the epithelium with PTEN?
Yeah.
And can you address this question doing conditional inactivation of PTEN?
Yeah. I mean, I would say I'd like to do a genetic experiment. I don't know for sure yet how it's going to pan out. We've been able to make mice that are knocked out for PTEN lung but still can maintain PTEN, and we're trying to work them up. We've had some problems breeding them with the first things that we tried to cross them with to do experiments have been sort of stymied, so we're going to have to try other models that we cross them with. But and also we're planning on doing some xenotransplants into them, or some syngeneic transplants in them to ask this question. But that's a really good question. I think there was such a controversy over the stromal mutations, and I've never looked myself.
I know, but maybe you put that back in the radar screen.
Yeah. It may be that we would see something, that it could be happening. I think it's possible. It's worth checking.
Ramon, I was just going to make a comment with regard to this correlation between the change in MYC message going down and MYC protein, or the other way around, MYC message going up and MYC protein going down. A possible explanation for that is that AKT, as you know, turns off GSK3. GSK3 phosphorylates MYC at serine 58, which creates an E3 ligase-dependent degradation of MYC. So, and in VMYC, consistent with that VMYC, that serine 58 is mutated to an alanine, so it doesn't get degraded.
Yeah.
So, when you inhibit Pik3 kinase, GSK3 could lead to MYC degradation, and therefore your result would be explained by that.
Our data would be consistent with that. We've done some I didn't go into it because it always confuses me and everybody else, the GSK3 regulation, but it's consistent with the idea that the Pik3 kinase inhibition is leading to a change in the GSK3 beta phosphorylation of MYC that would be leading to its degradation. And then we get this discrepant regulation of the mRNA in the opposite direction.
Well, that's because virtually everything you do in biology, when something goes down, right, whatever is supposed to make it go up goes up. So it's because MYC goes down, probably that the message goes up.
Well, I mean, it's nice that knowing that you can. It looks at least. I mean, and I don't. Elias has more data on this. He's also has knockdowns of MYC in combination with stable knockdowns of MYC in combination with the Pik3 kinase inhibitor to show that there is a real MYC effect. I don't want to say this is all based on these inhibitors, but I think what's really kind of cool about this is that we're trying to target two completely different pathways at the same time, and we're seeing some kind of efficacy. And actually, at first, I was really not expecting anything, and it's better than any combination that we've seen. So it kind of makes sense. That's what made these tumors was a MYC and a Pik3 kinase change. So it makes sense.
So actually, following Lou's comments, actually GSK3 has been shown to phosphorylate a lot of transstringing factor like c-MYC and c-JON for degradation. Mm-hmm. Therefore, Pik3ca inhibitor will lead to reactivation of GSK3. So now c-JON and c-MYC will be ubiquitinated.
And degraded, right?
Yeah, wait a minute. This process is very funny because Bill Tansey, previous investigator in Cold Spring Harbor, showed that ubiquitinated c-MYC are more powerful, more activated.
Yeah.
It's like a flower.
I remember that paper, yes.
Yeah, so therefore you have this kind of paradox that the ubiquitinated were in, but more active, so that explains everything. Yeah. C-MYC turn on c-MYC and c-JON turn on c-JON, but they were unstable, but meantime they're more active.
Yeah.
So that might be the
interesting
paradox.
comment. Yeah, thanks.
I mean, Ramon, I was interested in your architecture of the complex between PRX2 and PTEN, and you had the tail region of P10 binding to the IP4 domain, but not the PDZ domain, which is what you would expect.
Yeah, we had to resequence a lot of things.
Isn't that very surprising? Is that
It was very surprising. I have to say, it's what it seems to be.
Does that mean other sort of similar IP4 domains, do they also bind PDZ binding motifs, as people looked into this? Or?
Well, the only thing I can say is people have looked into PREX1's IP4P domain and its ability to interact with, basically, we haven't repeated this. Basically, there is evidence in the PREX1 literature that this IP4P domain can actually bind to the PDZ domain that's within PREX1. So for this auto-binding effect, and it's this PDZ domain is interacting with IP4, not the carboxy terminus of the IP4P domain, but within it. So there is some precedent for this PDZ domain to not behave like the classic ones, but to be able to interact with non-carboxy terminal amino acids. So that's the only thing I can say. PREX1, on the other hand, does not bind to P10 at all from anything that we've been able to see.
Okay, thank you very much, Ramon, for getting us off to a fantastic start.